Heredity explains why most family members resemble each other. Genetics is the study of heredity. Genes are the segments of DNA that carry instructions for a characteristic or a trait. Many of these genes code for enzymes. These enzymes control metabolic reactions that control how we look or what kind of metabolic reactions we can carry out. Genetics was first studied by Gregor Mendel in the mid-1800s. His work was shelved for almost 40 years because we had to figure out cell division before we could make sense of some of what he found out. 100 years later, the Human Genome Project started. Human DNA was scanned and we were able to determine human DNA sequences. Geneticists can now manipulate DNA, not only in the human genome but in a number of other genomes. This opened the door for genetic engineering. Chromosomes are the DNA and the associated proteins. These proteins help with controlling the DNA and packaging the DNA. Humans have 46 chromosomes, or 23 pair. 22 pair are known as autosomes. One pair is the sex chromosomes. These chromosomes determine the genetic sex of the individual. Chromosomes that carry genes for the same trait are said to be homologous, so the paired chromosomes are the homologous chromosomes. Your karyotype is the diploid chromosome complement. For humans, that's 46 chromosomes. The genome is the genetic makeup. It represents two sets of instructions. One set of instructions came from the mother, and one set came from the father. This is a picture of a karyotype. Here you see the 22 autosomes, and this is the sex chromosome. So this is a genetic female because we have two X chromosomes. The X chromosomes are the same. With an X chromosome and a Y chromosome, the Y chromosome would be much shorter than the X. Genes that code for the same characteristic are said to be alleles. We have paired alleles because we have paired genes. One allele for each trait comes from the father and one comes from the mother. Alleles are at the same locus or location on the homologous chromosomes. An individual is said to be homozygous if both alleles are identical. They're heterozygous if the alleles are different. A dominant allele will mask the presence of its partner, and the recessive allele is the allele that's masked by the dominant. The recessive allele will not be expressed in the phenotype. The genotype is the genetic makeup of an individual. Do you have two dominant genes for that trait, one dominant gene and one recessive, two recessive? That's your genotype. Your phenotype is the expression of your genotype. It's what you look like. It's what you can do. Your genotype would be two dominant genes or one dominant, one recessive gene. Your phenotype would be you display the dominant characteristic. Sexual reproduction allows for genetic variation in humans. This means that each individual is unique in their genetic makeup, unless of course you have an identical twin. Genetic variation is due to independent assortment of chromosomes, crossover of homologous chromosomes during meiosis I, and random fertilization. In this cell we have 3 pairs of chromosomes. So they have been duplicated and they're lining up, so we're going to go through meiosis and get our haploid cells. When the chromosomes line up during metaphase, the order is random. Notice here all of the purples are on one side, and then we have various mixtures of purples and greens on various sides. We have different haploid cells that we have gotten as a result of meiosis. Because this particular cell has 3 pair, the possible combinations or the possible different haploid cells we could get would be 2 to the 3rd power, or 8. And that's what we have, 8 different possible cells as a result of meiosis. In humans, 2 to the 23 is the possible number of combinations. That's over 8.5 million possible combinations. Some genes are linked, that is, they're found on the same chromosome, so they're always inherited together. During meiosis I, the homologous chromosomes line up on the equator, so that we have 4 chromosomes lined up together. This is sometimes referred to as a tetrad. Now in this particular instance, hair color and eye color genes are on the same chromosome, so they would always be inherited together. This particular chromosome carries recessive genes for both hair color and eye color, and this chromosome carries dominant genes for hair color and eye color. This would mean that dominant hair and eye color were always inherited together, and recessive eye and hair color were always inherited together. Well, because these chromosomes are so close together, the DNA may tangle and break, and the genes get rearranged on the chromosomes. So that what we end up is one chromosome that still has recessive everything, and one chromosome that still has dominant everything, but these two chromosomes are now changed. Now recessive hair color could be inherited with dominant eye color, and dominant hair color could be inherited with recessive eye color. This is completely random as to when this happens or how this happens. Any of the 8.5 million possible eggs could be fertilized by any of the 8.5 million possible sperm. That's part of what random fertilization means. If you do the math, that means there are 72 trillion possible zygotes that could occur. Add the variable of crossover, and even more eggs and sperm variations are possible. It's a wonder that families resemble each other at all. Add to this that in large populations, males and females mate randomly. There is no pre-selection of which male will mate with which female. There are several different types of inheritance. The first we'll look at is dominant recessive inheritance. This reflects the interaction between a dominant gene and a recessive gene. If parental genotypes are known, a Punnett square or Punnett square can be used to determine the probable genotypes of the offspring. An individual with one dominant gene will show the dominant characteristic. The recessive phenotype will only show if the individual has two recessive genes. So we will look at something called tongue rolling, the ability to roll your tongue. Here we have a female that phenotypically shows the characteristic but is carrying a recessive trait. The male has the same thing, shows the dominant characteristic but is carrying a recessive trait. So phenotypically these are both tongue rollers. If we do the Punnett square, we will find that three of their offspring, there's the probability that three of their offspring will be tongue rollers, and one of their offspring will be unable to roll their tongue. Now, because this is a heterozygote mating with a heterozygote, this phenotypic ratio of 3:1 is always going to show. This is an important ratio to remember as we start talking about recessive genes and recessive diseases. Here's a list of some of the traits that are inherited by simple dominant recessive inheritance. Tongue rolling we've talked about. Who knew astigmatism is dominant to having normal vision? Freckles, dimples, being able to taste that yicky chemical, having a widow's peak, or a double-jointed thumb are all dominant characteristics. Syndactyly is the webbing between your digits. Achondroplasia is a condition where there is not normal endochondral ossification. Now, this is not truly dominant recessive because you can see the heterozygote shows incomplete dominance. If the individual is heterozygous, they're a dwarf. But if they're homozygous for the recessive allele, then they really don't live long enough to even be born. Huntington's disease is also a dominant disease. Here are some recessive characteristics. These are only going to show if the individual gets two recessive genes, Tay-Sachs disease and cystic fibrosis. As we have just seen, genes control the presence or absence of certain diseases. Disorders caused by dominant genes are relatively uncommon. If a gene is dominant, as we saw with achondroplasia, then the individual would die as an embryo, a fetus, or a child. So lethal dominant genes would not survive to be passed on to their offspring. Some dominant conditions develop symptoms after the individual has reproduced. We call these delayed action genes. Huntington's disease is such a disease. In Huntington's disease, there's a degeneration of the basal nuclei that leads to a loss of muscle function. The symptoms don't start to show until about age 40. Genetic testing can be done to determine if the gene is present in the individual prior to them having children. Individuals that have Huntington's only need the one dominant gene, so their offspring would have a 50-50 chance of inheriting that dominant gene from them. Some individuals are being genetically tested and opting not to have children. Recessive disorders are much more common because you need to get two genes to show the trait. In this case, both parents have to be carriers. They're both heterozygous. They don't show the disorder, but they both carry a gene for the disorder. Albinism is one such disorder. Individuals who are albinos are unable to produce any form of pigmentation. Cystic fibrosis we've discussed a couple of times. In this disease, the individuals produce a very thick mucus that does a lot of damage in the lung, as well as having pancreatic enzyme disorders. Tay-Sachs disease is a disease where there is an enzyme deficit in the brain. A certain lipid is not cleared from the brain and builds up in the brain. Symptoms show before age 1 and it is fatal. Another type of inheritance is incomplete dominance. Here, neither gene shows in the heterozygote. The heterozygote gives you a different phenotype altogether. An example of a disease here is sickle cell anemia. Hemoglobin A is normal hemoglobin. This is the hemoglobin that easily picks up oxygen and transports it throughout your body. Hemoglobin S is a faulty hemoglobin. Hemoglobin S will crystallize when blood oxygen levels are low. This crystallization of the hemoglobin causes the cells to sickle. They become fragile and easily break, so you have a lot of hemolysis, and they can also block capillaries, leading to tissue ischemia. The difference between hemoglobin A and hemoglobin S is one amino acid in the 6th position of the beta chain of the hemoglobin molecule. Individuals who are homozygous for normal hemoglobin, hemoglobin A, have no problem. Individuals who are heterozygous, have some hemoglobin A and some hemoglobin S, have a problem only if they're under extreme conditions of low oxygen. For example, if they go up above 10,000 feet in an unpressurized plane. These individuals are said to have sickle cell trait. They seldom, if ever, have any indication that they have the disease. Individuals who are homozygous for hemoglobin S have sickle cell anemia. They have frequent crises with damage to tissue and anemia. Any type of low oxygen condition, like running up a flight of stairs, can cause them to have low oxygen in joints and leg muscles. This can cause the red cells in those areas to sickle, block capillaries, and cause ischemic damage to tissue. Fortunately, some drugs have been developed to treat this disease. Another type of inheritance is multiple allele inheritance. Some genes have more than two alleles that control the same trait. You still only get two alleles, but in the gene pool there's the possibility of several alleles. The ABO blood groups are an example of multiple allele inheritance. The genes for A blood and the genes for B blood are dominant to the genes that give you type O blood. Genes for A and B are codominant. There are 4 blood group phenotypes, O, A, B, and AB. The only way you can have group O blood is to be homozygous recessive for the recessive gene. If you have group A blood, there are 2 possible genotypes. If you have group B blood, there are 2 possible genotypes. If you have group AB blood, there is only one possible genotype, one gene for A and one gene for B. As you can see, group O is the most common in all populations. Group A runs second in all populations. Group B is getting a little more rare, and group AB is very rare. Another type of inheritance is sex-linked inheritance. Here the inherited traits are carried by genes on the sex chromosomes. The Y chromosome is shorter than the X chromosome, so it carries less genetic information. This means that males do not have corresponding genes for genes on the X chromosome. If males inherit a recessive trait on the X chromosome, it's going to display in his phenotype. Because females have 2 X chromosomes, they would have to get 2 recessive genes. Females are usually carriers. Sex-linked traits are things like red-green color blindness and hemophilia. Most phenotypes are not dependent on single genes. Most of them are controlled by several gene pairs that are at different locations on the genome. They're on different chromosomes. This results in a continuous or quantitative phenotypic variation between 2 extremes. That is, there is a gradation between a high and a low. Skin color, height, metabolic rate, and intelligence are all controlled by polygenetic inheritance. Let's look at skin color. We know that there are at least 3 separate genes for skin color on 3 different chromosomes. Dominant is to have dark skin. Recessive is to have light skin. If all of your genes for skin color are dominant, you have the darkest skin possible. If all of your genes for skin color are recessive, you have the lightest skin possible. The number of dominant and recessive genes give you a range of skin pigment. Looking at this example, we start with parents that have either all recessive or all dominant genes. In the first cross, the offspring will be half and half, so they will have that medium color skin. Now, if these offspring mate, we get this range of very light skin, darker, 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 and the darkest of skin. So this is an example of polygenetic inheritance, a range of values depending upon the exact genotype you get. Environmental factors can influence phenotype. Drugs taken during pregnancy can affect the fetus. We've already talked about thalidomide and how it prevented limbs from forming. Tetracycline, a relatively innocent-looking kind of antibiotic, can cause permanent staining in the teeth if the mother takes it while she's pregnant. Poor infant nutrition can mean that an individual does not reach their full genetic capability in height or intelligence. If you have problems with one gene, it can cause problems with the expression of other genes. For example, if you don't produce thyroxine, your metabolism rate will be very low. This would mean you would not provide sufficient energy for growth of other organs. This could affect height and intelligence. Individual choices can also affect your phenotype. People choose to smoke or drink excessive amounts of alcohol or do drugs. These are going to damage organs that genetically were probably programmed to last a lot longer than they're going to last as a result of this choice. People change their skin color by tanning. People may increase their muscle mass a little bit by bodybuilding. Besides the DNA we've discussed so far, there are also other extranuclear inheritance patterns. The protein coding genes, the one we've talked about so far, are what we consider the first level of information for inheritance. They compose less than 2% of the DNA in the cell. Second and third layers of information direct development of our genotype elsewhere. The other regulatory systems include small RNAs, epigenetic marks, and extranuclear or mitochondrial DNA. Small RNAs are part of the second layer of information. They were formerly believed to be the non-coding DNA in our genome, what was sometimes referred to as genetic junk. These genes only code for RNA. They don't code for a protein. These RNA-only genes act as mobile control freaks. They can act directly on DNA, on other RNAs, or on proteins. One of the things they can do is help tame aggressive jumping genes or transposons. Jumping genes are little bits of DNA that can jump around in your genome. As a result, they may jump into a gene and inactivate it, or jump into another gene and overactivate it. This can be damaging to you, so these small RNAs help control these. Small RNAs also control the timing of programmed death for cells during development. As we develop embryonically and fetally, there are many structures that are formed that are only temporary. They have to go away. These small RNAs help with making these structures go away. Mutations in these small RNA genes have been linked to several conditions, some types of lung cancer, some types of prostate cancer, and schizophrenia. It only takes a very few genes to build a human, but the complexity of the human is due to the small RNAs. As a result, these particular molecules are being heavily investigated. They're trying to use them to develop drugs to treat diseases that we have not successfully treated so far. Epigenetic marks are the third layer of gene control. This refers to the information that's stored in the proteins that bind to the DNA and in other chemical groups like methyl groups and acyl groups that bind to the DNA. Also, these are the proteins that are involved in packaging the chromatin. These chemical tags can help determine if DNA is available for transcription. They also account for the inactivation of one of the X chromosomes in females in the early embryo. Epigenetic marks may even predispose a cell to transform from normal to cancerous. Slight deviations in epigenetic marks may make the difference between an illness being incredibly mild or an illness being very severe in an individual. Genetic imprinting is one of the epigenetic marks. In most genes, maternal and paternal genes turn off and on at the same time. It doesn't make any difference which parent you got the gene from. But during gametogenesis, some genes are methylated. This is the genomic imprinted. These methylation tags tell the embryo whether they're from the mother or the father. The embryo reads the tags and will express only one gene. Imprinting is erased with each generation, usually, so that your imprinting is not passed on to your offspring. Mutations in imprinting may lead to pathology. Individuals who have Prader-Willi syndrome are mild to moderately retarded, short, and rather obese. Individuals with Angelman syndrome are severely retarded. They're unable to speak, they laugh uncontrollably, and they have jerky, puppet-like movements. Both of these syndromes are the result of a defect on the same gene in chromosome 15. The difference is epigenetic marking. If the gene that's expressed is the paternal gene, then Prader-Willi syndrome is the outcome. If the gene that's expressed is the maternal gene, Angelman syndrome is the outcome. There are 37 genes in the mitochondria. These genes are transmitted almost exclusively from the mother. The egg is really the only cell that contributes cytoplasm to the new zygote. There is a growing list of rare disorders that are being linked to mitochondrial DNA. Most of the disorders involve problems with the functioning of the mitochondria, the oxidative phosphorylation that occurs there. However, there are a few rare degenerative muscle disorders that have been linked to mitochondrial DNA and a few rare neurological problems. Some are beginning to link Alzheimer's and Parkinson's to mitochondrial DNA. We now have the ability to tell someone their genetic makeup. As a result, we can do genetic screening and counseling. One of the things we can do is identify the carrier state. Carriers will look fine, but they will be carrying a defective gene. We used to do this through pedigrees. We would take families and trace back through the families to see where the gene came from and who likely had the gene. Now we can do this with blood tests and DNA probes. Fetal testing is possible. Amniocentesis can be done at about the 14th week of gestation. Because you're sticking a needle through the placenta to withdraw some amniotic fluid, there is some danger to the fetus. Using ultrasound has reduced that danger. Chorionic villus sampling is a little safer and allows for testing as early as the 8th week, but it's recommended you wait until the 10th week. This allows you to check the genetics of the fetus. If there is a problem, then there is a decision that needs to be made about terminating the pregnancy. Amniocentesis, the decision has to be made very, very quickly. Chorionic villus sampling gives you a little more time to make that decision. Soon both of these tests will be outdated. There is a maternal blood test that will soon be available. Human gene therapy allows us to insert a functional gene into a defective cell. This has been tried for cystic fibrosis, muscular dystrophy, and severe combined immunodeficiency disorder. It's had limited success. Scientists are still working and are hopeful that one day they will be able to treat certain genetic deficiency diseases with gene therapy.